In section 3.3, we're going to continue looking at this question of whether supplements are truly beneficial to our bodies and to our health. And so in order to completely answer that question, we need to first look at membrane transport. So in this section, 3.3, we're going to split into a couple of different parts, but we're going to be looking at equilibrium, looking at the materials that are able to move freely through the membrane versus those that require a protein. We're going to look at the different types of transport, passive and active. We're going to look at different types of solutions like isotonic, hypertonic, hypotonic. And then at the end of this section, we're going to look at endocytosis versus exocytosis. So again, the question here is, are dietary supplements beneficial? Because one of the things that we know is that they don't rot like fruits and vegetables. So they last a little bit longer, but are they truly good for you? Well, in order to understand that, one of the things that we have to understand is that in order for substances to actually be beneficial, they have to get inside of your cells. So that means they're going to have to go through the plasma membrane. So this would include both food as well as substances. And then once these substances get on the inside of the cell, the reason that we say that's where they become beneficial is because nutrients that get inside the cell can then be used to make things like proteins or lipids. They can also be used to provide energy. So again, let's focus on this plasma membrane because this is what we already said is selectively permeable. So it only allows certain materials to come through. Now we know that hydrophobic substances move a lot easier because remember the inside of this bilayer are hydrophobic tails. And so hydrophobic substances typically move easier. Now again, why would substances move? Well, substances are all driven by a vocabulary term that we know as equilibrium. Because the only reason that substances would move is in order to get equal amounts of substances on both sides. Okay, so having equal amount of substances on both sides forms what we call as equilibrium. So what these substances will try to do is they're going to try to move from one side to the other in order to balance things out. So quick question is, what type of substances are allowed to move? Well, we have two categories here for substances, those that move freely on their own versus those that have to have a transport protein. So some examples of substances that move freely would include things like carbon dioxide, water, oxygen. All three of those are able to move in and out of the cell by themselves with ease. And again, the only reason they move is in order to balance out the amount of, say, carbon dioxide on the outside of your cell with the amount of carbon dioxide on the inside, or the same thing with water as well as oxygen. Substances that cannot move as freely, that have to have a transport protein in order to move them through, would be things like macromolecules, large molecules like carbohydrates, proteins, lipids, charged molecules, ions. So all of those things would have to have a transport protein. So we want to briefly look at two major types of transport that move materials in and out of the cell. In this part, we're going to focus on passive transport. So what makes transport passive? Well, the main thing is it does not require energy. You think about a person that you would describe as being kind of passive. Well, that would be a person that typically does not get worked up. They don't use a whole lot of energy to deal with stuff. So passive transport does not require energy. There are three types of passive transport: diffusion, facilitated diffusion, and osmosis. So what we want to do is look at the differences between these three types of passive transport. Well, in order to understand them, we have to first understand something known as a concentration gradient. So the word gradient just specifically means a difference. And so concentration gradient says that at any time there's a difference in the amount of a substance on one side of a membrane than on the other side. Remember, I told you the only reason that substances move is they're trying to get equilibrium. They're trying to get equal amounts of a substance on both sides of a membrane. And so if there's ever a difference, we say that there's a concentration gradient. So for example, let's say that you've got on one side of the membrane, you have 95% of a certain substance, but on the other side, there's only 90% of that substance. Well, obviously, there's a difference here. These two numbers are not equal. The difference is 5%. So we would say that that's our concentration gradient. There's more on one side than there is on the other. So with diffusion, diffusion is, again, a type of passive transport, so it does not require energy. It specifically is going to move substances from their higher concentration towards their lower concentration. So if you look at the image, for example, here you would notice that there's more carbon dioxide and there's more oxygen out here than there is in here. So diffusion says that these substances will move from their higher concentration towards their lower concentration. We would say that this is moving down the concentration gradient because it's going from high to low. Now, how long will these substances continue to move? Well, again, the whole goal here is to reach equilibrium. So both carbon dioxide and oxygen would move here until the amount of gases on this side are equal to the amount of gases on this side. Some examples of diffusion. Crystal light packet being dropped into a bottle of water. So you'll notice in the picture you put crystal light in to begin with. The higher concentration of crystal light would be here, but if you let it sit over time, the crystal light is going to diffuse from its higher concentration to lower concentration till the entire container of water has crystal light in it. Same thing with putting drops of colored dye in water or putting a tea bag in water. You would see that the tea bag or the dye would diffuse towards the lower concentration until the entire container of water had those materials in it. Perfume in the air. If you've ever noticed, if you spray perfume, to begin with, you only smell it in that one vicinity. But if you give it time, the perfume will diffuse through the air. It goes towards the areas of the room that don't have perfume in it until every aspect of the room smells like that perfume. Shaking salt into the water, lighting a cigarette and the smoke spreading, putting a sugar cube into a cup of coffee, all of these show a substance moving from its high concentration towards its low concentration until they get balanced out. Now, a second type of passive transport is known as facilitated diffusion. Facilitated diffusion is known as facilitated diffusion because it requires a facilitator. There has to be a transport protein in place to move these materials into the cell. Now again, it is passive, so it is going to not require energy, and it's going to move from high to low. But the key here is there has to be a transport protein in place to move these materials. A great example of this is how your cells transport glucose, the sugar. Glucose is able to move from a higher concentration towards a lower concentration, but there has to be a transport protein in place to carry it across, a facilitator, if you will. The third type of passive transport is known as osmosis. Now, the key with osmosis is that it's basically the same concept as diffusion, moving substance from high to low until it balances out without using energy. But the key here is what's actually moving. In osmosis, it's the water that's moving from the high concentration to the low concentration. And so anytime we talk about osmosis and the movement of water, we also have to discuss a subject called tonicity. Tonicity refers to the strength of a solution. And so there's three types of solutions with regards to tonicity that we discuss when we talk about the movement of water. Isotonic, hypotonic, and hypertonic. Let's look at the first one. Iso means equal. So in isotonic solution, you have the same amount of solute and water concentrations on the outside of the cell as you do the inside of the cell. And so where these two materials are the same or are equal, out and in, water moves out and in at equal rates, and so the cells maintain their same size and their same shape. So this would be the ideal solution or the ideal situation. But we know that situations are not always ideal. In some cases, we experience what we call hypertonic solutions. If you take a cell and you put it in a hypertonic solution, what that tells us is the concentration of solute, that's the stuff in the water, is higher on the outside of the cell than on the inside of the cell. Okay? So if the concentration of solutes is higher outside than in, then that also means that there's going to be more water in than there is out. And here's what we know about water. Water moves by osmosis, which says water moves from their higher concentration to their lower concentration. So if there's more water in the cell than there is out of the cell, the water will rush out, causing the cell to shrink. Crenation is what we call an animal cell that shrinks. Plasmolysis describes a plant cell that shrinks. Now, ideally, would you want your own cells to shrink? Well, obviously no. So this is something that you would want to avoid in your own body. You would not want red blood cells to shrink. However, this is something that we do to cells that we don't want to grow. So one of the preserving mechanism that we used for meat before we had refrigerators was to add salt to the meat. Now, the salt was not used for flavoring here. The salt was used to make a hypertonic solution to draw water out of the bacterial cells, causing them to die, to keep bacteria from growing on the meat. So we use hypertonic solutions in order to kill cells that we don't necessarily want there, but we want to prevent hypertonic solutions around our own cells. The third type of solution we see is a hypotonic solution. Hypotonic is the opposite of hyper. So a hypotonic solution is one that has higher solute content inside the cell, which means there'd be more water outside of the cell. And again, water tends to move from high to low. So if there's more water outside of the cell than there is inside the cell, water's going to rush in, causing the cell to swell. And in serious cases, it can actually lead to cytolysis, which causes the cell to burst, specifically for animal cells. So you can obviously see a problem here if your own cell were to be bursting. So it's definitely something you would kind of want to prevent for your own cells. But again, something that we use to get rid of cells we don't want. See, if you have a bacterial infection and you go to the doctor and they give you an antibiotic, what a lot of antibiotics do is they break down and put weak spots in their cell walls and in their plasma membranes. And so then if you drink plenty of fluids on top of that, you cause a hypotonic environment where water is going to rush into those bacterial cells, causing them to swell and to even burst, which would in turn kill the bacteria. So it's beneficial in that case, but again, could potentially be unbeneficial for your own red blood cells. If you add too much water to your red blood cells, you cause them to burst, and if the red blood cells burst, they can no longer do their job of carrying oxygen. So again, these different situations could potentially be dangerous for your own cells. Now in class, we're going to watch this animation, having a hard time getting it to work for video, so this will be something that we'll look at together in class.